This protocol allows the tracking and quantification of the degradation of a protein of interest in life and aging C. elegans. This in vivo and non-invasive technique requires only little microscopy setup to reliably convert dendra-2 into a bright and stable fluorophore.
The method is adaptable to mammalian system as well as zebrafish to study protein turnover within the proteostasis network as well as transport and trafficking. Take care not to provoke unwanted conversion of dendra-2 while scanning with the blue light laser, and to test and optimize the acquisition settings and conversion settings for each system and fusion protein.
Begin by growing age-matched nematodes at 20 degrees Celsius to the desired age for the experiment. On the day of the imaging experiment, melt general-grade agarose at a 3% concentration in double distilled water. While the agarose is cooling, cut the tip of a 1 milliliter pipette tip to facilitate aspiration of approximately 400 microliters of the melted agarose. Carefully add a few drops of agarose onto a clean glass slide, and immediately place another slide on top of the drops to create a thin pad of agarose between the two glass pieces. When the agarose has dried, carefully remove the top slide. When enough slides have been prepared, open the confocal microscope software and set the light path for excitation and emission for green dendra-2 at 486 to 553 nanometers and for red dendra-2 at 580 to 740 nanometers.